afternoon. Thanks for the introduction and for the invitation to speak to you today. So I'm going to be talking about the African genetic diversity and some of its applications in health. So the outline of the talk is I'm first going to describe why African genomes matter and hopefully convince you that African genomics is extremely important, not only locally, but also globally. I'm going to talk about some of the challenges in doing African genomics data analysis in terms of finding and analyzing the data. Then I'm going to talk a bit about a project that we did and it was published in Nature, where we studied African genetic diversity in some high coverage genomes and some of the implications for health. And then I'll talk a little bit about the projects that we're doing in H3A that are helping to provide tools for African genomic medicine. So African genomes matter to Africans, African individuals, because Africa suffers a disproportionate burden of diseases. This is both communicable and non-communicable diseases. And African genomes have a huge diversity. So they have shorter haploblocks, amazing diversity between different groups, even within a country. And this is because of the age of African populations and the fact that everybody else was derived from African populations. So this means that African populations have had a lot of time to evolve, and there's specific exposures or evolutionary pressures. And for example, there have been there's been evolution where there's been protection against malaria, but that same mutation has caused predisposition to sickle cell disease. There are also cases where there was a mutation that caused predisposition to kidney disease, but at the same time protected against trypanosomiasis. So African populations have evolved to have different susceptibilities to diseases and different SNPs and variants that are associated with their susceptibility to diseases. And then, of course, also this means that African populations and all worldly populations have different responses to drugs. So an example is efavirenz, which is a drug for treating HIV. So there's a variant in the CYP2B6 gene, which slows the ability to break down the drug. And if you get the incorrect dose and you have this variant, then you have terrible neuropsychiatric side effects. And in fact, if you look closely, 20% of the population in Zimbabwe and 13.5% of the population in Botswana have 2 copies of this allele. So what this means is that instead of using the traditional way of just treating everybody the same way, so you have a particular dosing for everybody, and then some people have these side effects, one could then do some genetic genotyping to see if these individuals have this variant, and then you can adjust the dose accordingly. This is moving obviously towards precision medicine. But in order to do this, you obviously need information about what populations have this variant and the effect of this variant. So that's why our African genomes matter locally, but they also matter globally. If you look at ancient history, so as I said, that the Africans are the origin of mankind, and so everybody else is sort of derived from African populations. And then you had this out of Africa migration. But then more recently, you've had slavery and additional migration, which has caused people of African descent to move to other countries. And then more currently, even you have a natural increase in immigration across different borders, and in many cases in Africa, this has been due to conflicts and adverse conditions in particular African countries. So what this means is that you have African individuals and people of African descent presenting at clinics worldwide. And so when they present, the clinicians need to be armed with enough information to be able to treat them appropriately. So the other issue with African genomic data is that the reference genomes have been developed using genomes of non-Africans traditionally, mostly Europeans. And when there was a recent study by Sherman et al. That looked at an African pan-genome, they identified, and that was of African-American individuals, they identified a number of novel regions that weren't represented in the reference genome. The other thing about African data is that the data can put other data for European populations, for example, into context. So is a SNP really rare just because it's been seen, hardly been seen in other populations? It could in fact be very common in African populations. And the African data can also improve our understanding of human diseases and evolution because this is the origin of populations. And the variants found in African genomes can help impact health in others. So one of the examples of this is PCSK9, which is a proprotein convertase subtilisin. This has been shown to be involved in LDL cholesterol. So when there are 2 studies that have reported mutations in this gene in studies that particularly looked at African populations. And they found that where there was a mutation, there was actually protection against heart disease. And so in the second study below, you can see there was 88% reduction in risk for coronary heart disease if you had this mutation. So it's a nonsense mutation that's found in this gene, and it was found almost exclusively in individuals of African descent. So subsequently, research has taken this gene and identified inhibitors to this protein. So in a way of sort of mimicking the loss of function mutations that you're seeing in some individuals of African descent, you can block this particular gene, and you get the net effect of lower LDL cholesterol and then reduced factors, risk of cardiovascular diseases. So this is one way where the information in African populations has actually benefited other world populations. But the problem is that there's a bit of a challenge in finding and analyzing African data. So African data, we know that non-African data is not always applicable to African populations because of the different, the way these different genomes have evolved and the diversity in African genomes. But African populations have traditionally been pretty much understudied in genomics. And it's also underrepresented, therefore, in public databases. And even when there is data, it's not always submitted to public databases. And this is because there's been historical exploitation of African samples. So there is a reluctance to share. Obviously, there's the importance of protecting the rights and privacy of participants. So you have to have the appropriate consent to share. But also the sharing hesitation is a little bit to do with inadequate infrastructure for data generation. So if you've had this huge expense of generating the data, you're really reluctant to share it with others, and also to analyze the data. So you want to be able to have the power to analyze the data before somebody else in the world scoops you. But just some illustrations of the underrepresentation. So in the Omics Discovery Index, this shows the amount of data from different world populations for different data types. You can see in all cases, the Europeans are completely overrepresented and the Africans are reasonably underrepresented. Not necessarily as much as the Asian populations, but when you look at the per population numbers, then it is pretty underrepresented. Looking at the pie chart on the right, this is the number of submitters to EGA by region, and extremely low proportion from Africa. And many of those are from South Africa. But note that this represents where the submitters are from, not necessarily where the data is from, but still, if you look at the data, it's very underrepresented in African populations. Likewise, the GWAS catalog is very low representation. This has increased more recently, but interestingly, then the percentage of associations observed in the data that is available for African populations is double what you would see in European populations. So it just shows the value of when you do get African data, that it really can give you a lot of information. But luckily, there is an increasing availability of African genomes. So this is published, this is the representation of different populations in exact and 1,000 genomes and other projects. This was in 2016. If you look at the stats now in 2021, this first chart, the first bar on this graph shows the African representation. It's African stroke African American, and actually most of it is African American, but it's showing an increase in those of African ancestry. But also more recently, there have been a number of studies or big papers that have been published where the representation of African genomes compared to non-African genomes has increased. So the blue here is non-African and the browny color is African genomes. And you can see that more recently, there have been some studies that almost exclusively focused on African genomes. So the one in red here is the one I'm going to talk about next. So this is a study that was done in the H3A Consortium. So H3A stands for Human Hereditary and Health in Africa. And the consortium is set up to look at the genetic and environmental basis for diseases in African populations. And through the various different projects that were developed, there was a need identified for a better tools for looking at African genomes and more African genomic data. So a number of genomes were sequenced, sampled from previously unsampled regions or ethnic groups, and that was to design a genotyping array. And then the consortium came together and did a full analysis of these high coverage genomes to gain some insights into African genomics. So I'll start with the acknowledgements on this paper. It big group of a collaborative effort across the consortium with some collaborators in the US who are all involved in the Africa Consortium. So it was a really an amazing example of an African-led and mostly African-run project. And so this involved genome sequencing, as I said, from underrepresented groups. So we have more than 2,000 ethno-linguistic groups in Africa, very few of whom have been sampled in terms of sequencing. So in this project, about 350 individuals from multiple ethnic groups were sequenced, and then this was added to sequence data from the H3A Consortium project, who did medium coverage sequencing. So in total, there were 426 individuals from 13 countries, representing 50 ethno-linguistic groups. So the ones in red here are those that are from the H3A. The green are from Trypanogen, and then there were some other samples that were used as comparisons. So the data were treated as you would normally treat an NGS. You do QC, alignment, variant calling, and then there was joint calling and refinement and improvement of the calls and the alignments. And then there were several different groups. So the big analysis groups split into teams, some looking at population diversity and ancestry, signatures of selection, rare and novel variation, and then medically relevant variants. So some used only the high coverage data, which was about 31 million SNPs, and some used both the high and medium coverage data, which was about 41 million SNPs. So just a quick view of the data. So this is the PCA showing how the different populations split. So there are 4 major language families in sub-Saharan Africa. There's the Afro-Asiatic, Nilo-Saharan, Niger-Congo, and the Khoi and San. And what's interesting is that it largely follows the map of Africa, but there is a split, for example, here in Uganda, where you had 2 distinct groups, depending on whether they were Nilo-Saharan or whether they were more the Bantu-speaking groups. So first, I'm not going to cover all the projects because there will be a talk about later from Ananya who will cover some of the analyses. So I'll just cover the selection section and the medically relevant variants. So in selection, the group did looked at the distribution of composite likelihood ratio statistic scores for 10 kb windows across the genome. They identified 107 outlier selection signals, which were at a p-value of less than 0.001. And of those, 62 were novel that had not been identified before in African genomes. And when they looked closely at these regions, I found that there were probably adaptations to local environments, diets, or pathogens. So they were linked to genes involved in viral immunity, DNA repair, and metabolism. So this just shows you some of those genes. There was also a variation in the selection signals between different populations that were under study. So here you can see a bar chart of just a few of those genes that were studied in detail. And you can see that there's different significance levels for the different populations. So this is definitely a distinct variation across the continent in these selection signals. What was also interesting was that the non-coding selection loci converged on GWAS hits. So there's 36 regions with at least one significant GWAS hits, and they map to 24 different disease traits. And there were also 5 non-coding regions. 5 of these non-coding regions had hits in 2 or more populations, so they weren't just population specific. Some of them also were enriched for tissue eQTLs, particularly in blood, skin, and vascular tissues. And generally, these were consistent across populations except Botswana. So Botswana was the group that actually had the highest number of novel SNPs and was quite diverse from some of the other populations studied. And some of the regulatory loci that were identified were involved in chronic kidney disease, uterine fibroids, and various blood red cell activities. So then there was an investigation into the putatively damaging variants. So it looked at loss of functions. And so these include canonical splice site, stop gain, stop loss, etc. And they found that non-singleton potential loss of function variants are shared across populations. So the red here shows those that are shared across all populations, whereas the other colors are a bit more population specific. They also found that these converged on disease genes. There were a number of disease genes identified with these potential loss of function variants. Interestingly, some of these were linked to infectious diseases. So the group looked at the correlation between influenza mortality rate from WHO figures and the ratio of putative loss of function variants in direct versus indirect influenza-associated genes. So this was taken from gene cards, looking at expression data. And there's a moderate negative correlation was identified between these two factors. And this was particularly evident in West African populations, which was not identified in the more southern populations. The gray just shows the background of 1,000 permutations of random genes. Then we looked at African genomes that were medically relevant. So first of all, looking at the ACMG list, this is American College of Medical Genetics and Genomics. So they have a list of actionable genes. And interestingly, these were uncommon in the data set that we were looking at. So there were only 8 ACMG variants found in 8 individuals. And these are the different genes that those were found in. Contrastingly, in ClinVar, when we looked at the likely pathogenic variants that were identified as such in ClinVar as sort of level 5, these were actually commonly observed with about 2 to 14 per person, per individual in the study group, with a median of about 7. And and many of these were in at least one population. So it means that there's a number of African individuals walking around with a reasonable burden of ClinVar pathogenic variants. This again was population variable, so some of them were more prevalent in some populations than others. So if we looked a bit closer at some of these medically relevant variants, you could see that there was a lot of variation depending on the region and the ethnic group that it was studied in. So these are just some of the interesting ones that the medically relevant group looked at, because these happen to be known to be relevant in African populations. So of interest was the splits with the 2 Ugandan populations. And this is where, if we go back to the PCA, you could see those, even though it's in the same country, there were 2 distinct population clusters for this group. And you can see that they have very different distributions of these allele frequencies of these particular variants. And another interesting variation was this HLA-B star 5701, which is associated with sensitivity to Abacavir, which is another HIV drug. And you can see that this was prevalent in the one group of Ugandans, but not at all, completely absent from the other. And interestingly, the ones that are relevant to malaria adaptation, like HBS, which is the sickle cell gene, and APOL1 for trypanosomiasis, which is also sort of in the malaria belt, you can see that those are prevalent in West Africa and in the malaria belt, and almost absent from absolutely absent from Southern African populations. The Ugandan one, the UNS, you can see that the hemoglobin is absent from there. And originally it was thought because malaria only came into the East Africa, the far eastern side a bit later, but there's now potential evidence that is coming from North African population migration, and having more North African populations in the data set would help to improve our understanding of that process. Another gene that was looked at was GCPD deficiency. So these also protect against severe malaria, and there was a lot of variation in these. These are a number of different variants in the alleles in the G6PD gene, and it differs with both the population and with the gender. So you can see that there's some are more prevalent in the females compared to the males. The absence here of females is simply because there were no females in that data set. So that's just an artifact. But you can see that there's a lot of diversity in these medically relevant variants, both in terms of where they are in the population, in terms of the geographical location, and that's often linked to things like, their location, like a malaria belt or other environmental factors. So what is the implication of this and other work in African genomes? Well, what is demonstrated is that individuals carrying likely pathogenic variants are reasonably common in African populations. And so we need to revisit this annotation in databases like ClinVar. The disease variants in African genomes need to be further curated to improve our understanding of their distribution, because this is just a small study, but we need to look a bit wider across the continent. And there are many novel SNPs still to be discovered. So the study identified 3.4 million novel SNPs. Most of them came from the newly sampled groups. And 2 to 5% of all these SNPs were in each population, so they were in all the populations. So if you look at the graph showing, if you just add the number of SNPs added for each new population, starting with Botswana, which had a large variation, you can see that there's still an increase with novel SNP discovery. And if you're looking at common SNPs, it starts to plateau a little bit, but still between these last 2 points, it's about 6,000 novel SNPs. So it means there's a lot of new SNPs still to be discovered. The other thing to further explore was other variants like copy number variants and structural variants, and there are projects underway to do that in these particular genomes. There are other regions in Africa that's left to be explored. There's North and Central Africa were missing from this data set entirely, and there are efforts underway to improve that. We also don't have enough data yet or evidence for broad genomic medicine applications. So this was a small data set, but we need more data with additional phenotype data linked to it to inform genomic medicine. So we need to pull together all the African data that does exist and build better tools, and also to improve the infrastructure for genomic medicine, like how do you identify these, how do you build the evidence, what is the actionability, how do you determine that, how do you generate reports once you've done whole genome sequencing. There's a whole infrastructure around genomic medicine that has to be developed. So H3Africa and H3A have done quite a lot in this space, and a lot of their work can be leveraged. So H3Africa, for example, is building evidence for genotype-phenotype associations across multiple different diseases. Together, H3A and H3Africa have built enormous research infrastructure and clinical facilities through these projects, because they've had to collect cohorts and have the research, the infrastructure to do the research. There's a huge training program across these across the consortium to build relevant skills for data analysis and interpretation. So H3A has trained over 4,000 unique individuals across Africa in the last few years in various aspects of data analysis and interpretation. We also are ensuring that there is access to computing infrastructure. So once you start doing whole genome sequencing, the data sets get extremely large, and you have to have reasonable infrastructure for transfer, processing, and storage. So we've been developing containerized workflows to have reproducible analysis of the data. We provide support for data analysis. We've developed, together with the H3A Consortium, standard case report forms, so a standard way of collecting phenotype data and other clinical data. We do the preparation of submission to the EGA to make sure that the data actually goes into public repositories and is fair, findable, accessible, interoperable, and reusable. And we're also developing some portals called AGPD and Genomic Medicine Portal, which I'll talk about now. So these are the resources. So we're building tools and resources for, through all of this, we're building tools and resources for enabling genomic medicine. So one of these tools is the African Genomic Medicine Portal. So the reason we wanted to develop this was that if you, in my personal experience, I was trying to go to different databases and find information about African populations for variants that might be of medical importance. And so you go to PharmGKB, you go to DisGeneNet, and actually finding the African data is really difficult because it's just labeled sometimes as African or African American, and there's no granularity in terms of what country in Africa it comes from, and you have to go to these multiple different places to find this. So we wanted to develop a portal that was focusing particularly on African variants and information that could be relevant to genomic medicine or some sort of medical application, but it's not. As a disclaimer, it's not supposed to be a clinical tool. This is a research tool for finding information that might be relevant. And so we wanted to incorporate disease and clinical drug and genetic research from various different sources. So the project lead is Samar from Egypt, and she's got a great team of developers and curators. So the way the portal is developed, so it's the data is extracted from PharmGKB and DisGeneNet. So we started extracting from other resources, but DisGeneNet turns out has all those resources in it. And there's a lot of data extraction from the literature as well. There's some filtering, there's manual curation, and a bit of text mining, and then the curated data goes into a portal. And the portal is a Django application, and it allows for various different kinds of search functions. So you can search by variant, by disease, by drug, or by gene. This is just the homepage. We're still designing it, so this is just a prototype. It has a tutorial and a frequently asked questions to provide some user support. And then, as I said, you can search by these different options, so disease, drug, variant, and gene. So what I did here was I searched for efavirenz under drug, and you can find it in Drug Bank. And then if you click on the link here, variants associated with this, you will then get the African-specific information for this particular drug and variants associated with it. So you can see that there's data from South Africa, Rwanda, etc. And we've also included African-American data in here. So it's a way that you don't have to go to PharmGKB and search and identify African and then go into each entry to see which part of Africa it's from. I try to summarize that all here. You can also search by a particular gene, and once you've got that gene, then you can get disease-associated information for that gene. So just picking up on the G6PD again. So this portal is available, and it's going to be available. The prototype is available, and it will be released later this month, we hope. So we hope people will test it. But what if you want to then find out more information about a variant in African population? So this just provides curated data from public resources. But we get many cases where people are doing like a rare disease study, or they're interested in one of these genes or variants, and they want to find out specifically what frequency this is found in African populations. So another tool that we've been developing is called the African Genome Variation Database, or AGVD. This is being led by Aiton Menkes, and the development of this interface was done by Davis Tott as part of his master's. And then the backend's being developed by Mohammed Al-Siddiq, but there's a big team of curators and other developers in this project. So you can search by a variant. You can get a variant view, for example, here, and it gives you links to some existing databases. So if there's a Cosmic link or a ClinVar link, for example, and this will link directly to the AGMP as well. You can then, so this is the detailed information for variants. You can do a lot of filtering, so you can filter by particular population groups, particular variants, diseases, and then you can get frequencies for these different more fine-grained level frequencies that you would get in the public databases. And then you can also view this alongside the public databases like Nomad, the 1,000 Genomes, Exact, etc. So we're not trying to reinvent the wheel and display exactly what they display, only we're trying to show their data next to a more fine-grained level analysis of African data. And then this can also be visualized on a world map. And so you can see the relative frequencies of a particular variant in different world populations. And then we've also developed, so those are two portals we've developed. We're also developing actual research tools. So we developed the H3A chip and an imputation service. So the imputation service uses the Michigan front end. So the data that was used in the Nature publication has been, we've created a reference panel for that, for which you can do imputation. So that's high coverage data imputation. And so it uses the Michigan front end and then generates reports using the same code. We've also done, we're also working on two other projects, a reference graph and a pan-genome. So this is to take some of the high coverage data that we have and build a pan-genome to identify novel regions, because the previous pan-genome approach was done on African Americans. And also to build reference graphs so that we can create an African reference graph for which you can then align sequences and call variants. And then just to finish off with a couple of other comments, we're looking at tools for federated analysis of data. So this is the H3Africa. I'm not going to go into this in any detail because Memano will be talking about this. There is an ecosystem within H3Africa. We've got data and clinical data and genomic data going to EGA, etc. And so we've got we're one of the driver projects for the genomics, the GA4GH. And so we're looking at building and including many of their standards. We have already done some of those, and also in generating a beacon, which is a way to search variants using the GA4GH beacon. And then another project, which again will be described in another talk, is the Common Infrastructure for National Cohorts in Europe, Canada, and Africa. And this is to basically harmonize the data, build tools, including authentication and authorization, to enable users to not have to move data. So you can have data in federated different places, but be able to compute across multiple cohorts while still maintaining the protection and the privacy of the individuals whose data there is. So to conclude, there are very many challenges in availability and accessibility of African data because of the reluctance to share, but it has enormous potential for novel discoveries. That paper that I presented just shows just the tip of the iceberg for the novel discoveries that we can find. And these novel discoveries and this information is required to really implement genomic medicine in Africa. And we've started to build these, but there's still a long way to go in terms of moving into the clinic and into full translation. We need more data generation, analysis, interpretation, and application to be able to derive these health benefits. So I'd like to acknowledge the H3A Consortium. These are just the work package chairs and co-chairs that I've mentioned here, and the team at UCT, but there's a huge consortium of over 200 members who make enormous contributions to all the projects in H3A. And thank you for your attention.